Hi, in this video we'll be looking at carbs and sugars. This is part of the biological molecules topic for AQA A-level biology. Okay, so let's start. As an introduction, we need to learn uses of carbohydrates. Carbohydrates have many uses, but two main uses are that they, first of all, can be used as respiratory substrates and energy storage. For example, starch, which is a store of glucose and energy for plants, and glycogen, which is a store of glucose in mammals. Some carbohydrates are also structural substrates. An example is cellulose, which is used in plant cell walls, which provides structure and support for plants. So now let's move on to the main content. Monomers. Monomers are small basic molecular units. Monosaccharides are a monomer and they are simple carbohydrates and simple sugars. Three examples of monosaccharides you need to learn are glucose, galactose, and fructose. Now that we've gone through monomers, we can move on to dimers. Dimers are two monomers which are bonded together. Disaccharides are dimers and they are also sugars. For example, maltose, lactose, and sucrose. Now we learn about how dimers bond together. This is through condensation reactions. Condensation is the joining of two molecules by the removal and release of water. Above this definition, I have written three different examples of condensation reactions which form these disaccharides. And I would advise that you learn these. Glucose plus glucose makes maltose and water. Glucose and galactose make lactose and water. Glucose and fructose makes sucrose and water. It would help if you probably learn ways to memorize this, because for some people it would be hard. And one way you could do this is, because we know there's glucose on both sides, there's glucose on all three and water on all three, we can focus on these. So for this, the order would be G, G, F, M, L, S. This is just a simple note that I write down in my brain when I try to remember these. Now, we move on to a visualization and a visual representation of the condensation reaction. Here we see two different glucose molecules, as seen here, undergo condensation reaction, which is the removal and release of water. Therefore, we'd expect water to be on the products side. We see how a H from the OH group here and an OH from the OH group here, they are lost and it forms a H2O on the product side. And through this, a bond called a glycosidic bond, which is a bond between sugars, this forms. It forms between the carbon, and the oxygen. Now that we've gone through condensation, we must try to establish what the opposite of this is, because condensation is essentially the joining. But what would the splitting be? The splitting would be hydrolysis. And the definition is hydrolysis is the splitting of a dimer or polymer by the addition of water. Or it could be the splitting of a molecule through the addition of water. Therefore, instead of water being on the product side, which is here, they will be on the reactant side. Here we see an example which is maltose and water, which makes two molecules of glucose. Here we see maltose and we see the glycosidic bond. The glycosidic bond will be hydrolyzed by a molecule of water, and in turn there will be two glucose molecules which form. And we see how here there's an OH group and the H group has been added. So essentially it's basically the opposite of the reaction we did here. Now let's go to specifics of glucose. As I said before, glucose is a monosaccharide. Glucose exists as two different types, alpha and beta. These signs that you see are the Greek alphabet, which is alpha and beta. Glucose is a hexose sugar. The prefix hex stands for 6, and therefore we know 6 carbons are present. Hence why the formula for it is C6H12O6. Now that we know what type of sugar it is, we need to learn the structure. So as I said before, there's beta and alpha glucose. This would be an example of an isomer. An isomer is where there's the same molecular formula but different structural formula, as we see beta glucose and alpha glucose. In beta glucose, we see how the OH group is at the top and how the OH group in alpha glucose is at the bottom. Beta at the top, OH, and alpha, the OH is at the bottom. Now, a key advice would be to learn the structure of glucose. Here it is alpha glucose. However, you can switch the OH and H around to make beta glucose. It might take some time to learn this structure by heart. However, I would advise to draw this structure over and over again, and this could make you memorize the structure easier. Now we move on to naming carbons. As we know, there's 6 carbons in glucose, so therefore we need to know which carbon is where. Therefore, we give them numbers. And these numbers help out with bonds later on, so we know which carbon reacts with which carbon of which molecule. So now, when we name carbons, we name them 1, 2, 3, 4, 5, 6, obviously. Carbon 1 will start here, carbon 2 will be here, carbon 3 will be here, carbon 4 will be here, and carbon 5 will be here, and carbon 6 will be here on the CH2OH. Sorry. Now we go to bond naming. And now that I've established which carbons are where, we can name bonds in a very specific manner. For example, a 1,4 glycosidic bond between a maltose. So first we can do bond naming by seeing where the bond had occurred. So the bond in this case had occurred on this lovely area. And now we can see which carbons took place in the bond. When we zoom in here, which carbon is it? Yep, it's carbon 1. And here, which carbon takes place? Yep, it's carbon 4. So therefore, we say that a 1 to 4 glycosidic bond has occurred. This is the end of the first episode on carbohydrates and sugars. The next video will look at polymers and polysaccharides, and also look at testing out sugars and starch. Thank you for watching this video, and I hope this video was found helpful. Thank you for watching.